Today, let us start with Chapter 5 Anatomy of Flowering Plants. First, tissue. The term tissue was given by N group. Group of cells having common function and common origin form tissues. Types of tissues. There are two types of tissue: meristematic tissue, permanent tissue. First, let us discuss about meristematic tissue. Meristematic tissues are those which retain power of division. They are thin-walled, small, and have dense cytoplasm. Cells of meristematic tissues are isodiametric, without intercellular spaces, and has large nucleus. In meristematic tissues, the secretory substances or ergastric substances like gums, resins, etc. are absent. Plastids are absent, but cluster of plastids known as protoplastids are present, and reserve food material is also absent. High food supply is present, and they have abundant mitochondria, and the abundant respiration takes place. Meristems are of two types with respect to origin and position. With respect to origin, they are of two types: primary, secondary. With respect to position, they are of three types: apical, intercalary, and lateral. First, let us discuss meristem with respect to origin. Primary meristem and secondary meristem. Primary meristem. They are derived directly from the cells of embryo or one of its derivatives. They are present from the beginning of the plant. So they are primary in origin. They help in growth of length in plants. Now, secondary meristem. They derived from the permanent cells, the tissue which is formed later during the development of the plant, which helps in growth of thickness or width of the plant. Now, let us move on to the other type of meristem with respect to position. First is apical meristem. Apical meristem is of two types: shoot apical meristem (SAM) and root apical meristem (RAM). The function of apical meristem tissue both in SAM and RAM. First, one cell undergoes mitosis to form two daughter cells. In two daughter cells, one cell have capability of dividing and other do not have capability. The one which is having capability, it further divides to form other two cells. Then one which is not, which do not have capability, they form permanent tissues and accumulate to increase the length of the root or shoot. Then intercalary meristem. The meristematic tissues present between two permanent tissues is called as intercalary meristem tissues. Intercalary meristem tissues may be present above the root, example grasses; at leaf base, example grasses; then below node, example mint. These also results in formation of primary permanent tissues which are helpful in growth of length in plants. Intercalary meristem tissues are separated tissues of the apical meristematic tissues. Now let us move on to the lateral meristematic tissues. They are present in the sides of the plants. These help in increase of thickness or girth of the plant. All lateral meristems are secondary in nature except one lateral meristem, that is intravesicular cambium. So types of lateral meristem. First is fascicular vascular cambium, which develops within the vascular bundle, so called as intravesicular vascular cambium, which is primary in origin. Then intervesicular cambium, which develops between the vascular bundles, known as medullary rays. Then cork cambium, it forms cork tough protective material during the secondary growth. Intravesicular cambium is only primary meristem which forms the secondary permanent tissue. Overview of meristems. First, apical, intercalary, and lateral. Apical is found as stiff tips of the plant, then intercalary intercalated between the tissues, then lateral on the sides. The apical meristem forms primary permanent tissue. It forms intercalary forms primary permanent tissue. Lateral forms secondary permanent tissue. Apical helps in elongation of growth. Then intercalary also helps in elongation growth, whereas lateral helps in diametric growth. Organization of apical meristem. First is shoot apical meristem. Here there are two fang-like structures which are derivatives forming leaf primordia, which are young leaves, and axillary buds. Axillary buds are of two types: vegetative bud and floral bud. Vegetative bud forms the branches, whereas floral bud forms the flowers. And here you can see the meristematic activity area, where it gives basally for the linear growth and accumulate the permanent cells here accumulate and helps in elongation growth. And here the narrow region shows the nodes and the broader region shows internodes. First, the theories given in shoot apex are: first is histogen theory, which was given by Hanstein. He states that there are three layers present in it: dermatogen, peribulum, pleurum. Dermatogen is the outermost layer which forms the epidermal tissue system. Then peribulum is the middle layer which forms cortex endodermis. Then pleurum is the innermost tissue which forms steels, which consists of PVP, that is pericycle vascular tissue, pith. Then tunica carpus theory, which was given by Smith. The shoot apex is divided into two types. Tunica, other is corpus. Tunica is single layer, small cells. It undergoes anticlinal division and it forms epidermis. Then corpus is it is of several layers which have large cells which undergoes periclinal or anticlinal division, where it forms ground and vascular tissues. Then root apex. In root apex, the first theory was given by Histogen theory, Hanstein. It is same as the shoot. It is same as the shoot apex theory. It has three layers: dermatogen, peribulum, and pleurum. Dermatogen forms epidermal tissues and root cap. Here peribulum forms cortex endodermis. Here steel forms PVP. Then corpor kappa theory given by Scout, similar to tunica carpus theory, where kappa acts as tunica and corpor acts as corpus. Here kappa forms the epidermis and root cap, and corpor forms the ground and vascular tissue. Last theory in root apex is quiescent-centered theory, which was given by Cloves. It is also called as waiting meristem theory, as it only works during the injury. When any mechanical injury occurs, the cells which do not have capability, they regain the capability of division and divide. So it is known as waiting meristem theory. Then secondary meristem. Secondary meristem is always laterally arranged, so it is called as cambium. It forms secondary permanent tissue. It increases the grip or diameter of the plant. Secondary meristem is present in dicots as well as gymnosperms, but absent in monocots. Example, let us see the stem of dicot. Here you can see the xylem, phloem, then interfascicular cambium, which is primary in origin, and interfascicular cambium, which is secondary in origin. These both together form vascular cambium. The interfascicular cambium, cork cambium are secondary meristems, whereas intrafascicular cambium is primary origin. Root. Here vascular cambium, cork cambium are secondary meristems. The permanent tissue or mature tissues. They are living or dead cells which are derived from the meristematic tissues, but they have lost the capability of division. They are known as permanent tissues. They have become mature, attaining a defined shape, size, and function. With respect to origin, the permanent tissues are of two types: primary permanent tissue, secondary permanent tissue. In primary permanent tissue, they are derived from the apical meristem and intercalary meristem, whereas secondary permanent tissues are derived from the lateral meristematic tissue. Composition. It may be simple or complex. In simple composition, one kind of cells are present. They perform similar function. For example, parenchyma, collenchyma, sclerenchyma. Whereas in complex permanent tissues, there are more than one type of cells. All cells work as unit. For example, xylem and phloem. Now let us discuss the characters of simple permanent tissues. Characters: parenchyma, collenchyma, sclerenchyma. Living or dead. Here living, dead. Cell wall, it has thin cell wall and it is made up of cellulose. Here thick, and it is made up of cellulose and pectin. And here thick, it is made up of lignin. Shape, it may be spherical, oval, polygonal, or elongated. It may be spherical, oval, polygonal, where it has several shapes. Then intercellular spaces, it may be present or absent. Here absent. Then division, capability of division present, nearly absent. Then refractive index, which is low here. Collenchyma has high refractive index because of pectin, which is shiny layer. Then sclerenchyma low. Chloroplast, present or absent. Occurrence, it is most common tissue which forms major component of organs and most abundant. Collenchyma is below epidermis, hypodermis. In dicots, it is present in leaves and stems, whereas absent in roots. where in monocots it is not present. And sclerenchyma is seen in fruit wall, seed coat, pulp of fruit, etc., which is hard. Now let us discuss about types of parenchyma. Chloroplast containing parenchyma known as chlorenchyma, which functions synthesis of food. Then secretory substances, secretory parenchyma, which secrete oil, nectar, etc. Then air spaces is known as aerenchyma, where hydrophytes used in biology and present in petiole of banana and canna. Water, mucilage and food storage parenchyma, example potato, tuber, aloe vera, opuntia, etc. Types of collenchyma. Thickenings of pectin is only present in corners, so it is called as angular collenchyma. Example marigold. If thickenings are in the form of plates, such type are known as lamellate collenchyma. Example sunflower. If thickenings are seen in intercellular spaces, called lacunate collenchyma. Example calotropis. Functions of collenchyma. Synthesis of food, mechanical support. They are peripheral and elastic. It allows flexibility to organs. They protect them from tearing, which can be caused due to the wind. It provides mechanical support to growing plants of the plant, such as young stem and petiole of a leaf. Then types of sclerenchyma tissues. Sclerenchymatous tissues are of two types: sclerites, fibers. Sclerites and fibers character is difference between the characters. Shape: sclerites have rod, star, oval, spherical, whereas fibers have elongated. Then thickenings high, less. Lumen is narrow, here less narrow lumen. Branching is in branching it is branched, it is unbranched. Groups, it may be single or it may be in single or groups. It is always in the groups. Now let us see types of sclerites. Here brachyscherites, stone cells. It is present in guava, pears and sapota, etc. Then macroscelites, which are rod cells, which are rod in rod shape. They are found in seed coat of legumes, example peas, beans, etc. Then osteosclerites, which are in bone shape or known as crop cells. They are found in seed coat of monocots of legumes. Then astrosclerites, which are star cells, which are in star shape. They are found in petiole of hydrophytes, nymphia like Nymphia lotus, and leaves of tea. Then trichosclerites. These are found in hydrophytes and aerial roots of monstera. Functions of sclerenchyma. They are chief mechanical tissue. They provide strength and rigidity to the plant. Now let us discuss about complex permanent tissue. Complex permanent tissue are of two types: xylem and phloem. Xylem, the term given by Nageli. It provides mechanical support and transport waters, which consists of four types of cells: vessels, tracheids, xylem parenchyma, xylem fibers. Vessels, they are dead cells with thick cell wall and absent in gymnosperms. Tracheids, they are dead with thick cell wall which are present in gymnosperms. Then xylem parenchyma are living cells with thin wall and present in gymnosperms. Xylem fibers are dead and have thick cell wall and present in gymnosperms. Tracheids, the single cell elongated tube-like cells with tapering ends are known as tracheids. They have thick wall made up of lignin. Tracheids give mechanical support as well as conduct water. Types of tracheids with respective thickenings. Here there are two types of primitive thickenings and advanced. In primitive we can see annual thickening, spiral thickening. In advanced we can see scleriform, reticulate and pitted thickenings. Then vessels, a long cylindrical tube-like structure made up of many cells like short, wide and thickened cells called vessel elements. The gradually cross walls are completely dissolved or perforated, forming in an elongated tube. And thickenings can be several types, but most common type is pitted thickenings. Types of wood. There are two types of wood: non-porous wood and porous wood. Here vessels are absent, seen in gymnosperms. In gymnosperm, plants like nettles do not show vessels. Nettles are like Neetum, Iftera, Welwitschia, etc. Here porous wood vessels are present, seen in angiosperms, but it is not seen in some angiosperms like Winteraceae, Tracheaceae, etc. Let us discuss about wood or xylem parenchyma. The living tissues storage is seen like food, starch or fat. Then radial condition of H2O by ray parenchyma. Lateral conduction is seen. Then xylem fibers or wood fibers. They are made up of sclerenchymatous cells and may be septate or non-septate. Here you can see the structure of xylem fibers. Types of xylem with respect to origin. First is primary and second is secondary. Primary during formation of primary body parts, and it is seen in procambium. Then here secondary, it is formed during the secondary growth. Example, cambium. Procambium is of two types: protoxylem, metaxylem. The first form, primary xylem, develops before the plant organ has completed its growth. Then metaxylem, later formed primary xylem, develops when the plant growth has completed its growth. Now let us differentiate the characters between the protoxylem and metaxylem. Trachea elements present. Size of elements, here smaller and narrower, larger and broader. Then thickening, primitive, annual and spiral seed. Then here advanced, remaining 3 like. Then fibers, here absent, here present. Now let us discuss types of primary xylem depending upon the position of protoxylem in related to metaxylem. Exarch and endarch. Exarch protoxylem. Here protoxylem is towards outside of the plant organ. The roots of monocots as well as dicots, we can see them, which have centripedal arrangement. That is, smaller are inside and the larger ones are outside. That means, then endarch, here protoxylem is towards inside. It is seen in stems of monocots and dicots, where we can see the centrifugal arrangement. Secondary xylem well differentiated into two systems: the axial or vertical system, then ray or horizontal system. In axial vertical system, we can find vessels, trachea, fibers, axial parenchyma. In ray or horizontal system, ray parenchyma. Axial or ventral system provides, they carry water in a vertical manner, whereas these ray or horizontal system, they supply water in a horizontal way. Then phloem, the components of phloem and characters between them. Sieve tubes, living, it has thin cell wall and functions like food transport. Phloem parenchyma, which is living and has thin cell wall, which helps in food storage. Companion cells, which are living, which have thin cell wall and help sieve tubes for food transport. Then phloem fibers, which are dead cells, which have thick cell wall made up of lignin, which are helpful in mechanical support. Then components of phloem in angiosperms and gymnosperms. In angiosperms, the phloem consists of sieve tubes, companion cells, parenchyma, phloem fibers. Whereas in gymnosperms, sieve cells, then albuminous cells, parenchyma and phloem fibers are present. Now let us discuss about sieve tubes, which are multicellular. They are formed by union of several thin-walled sieve tube cells or sieve elements or sieve tube members. They form elongated tubular structure called sieve tube. Cross wall of sieve tube have small pores due to presence of pores and walls are commonly called as sieve plates. Sieve plates connects the protoplasm of adjacent sieve tube members. The mature sieve tube has peripheral cytoplasm, a large vacuole and lacks nuclei. Under unfavorable condition, the pore get closed with carbohydrates temporarily called as callose plex. Here you can see the sieve plates which have pores which allow the water to pass through. Phloem proteins in sieve tubes, which help in movement of material, healing, then blockage of sieve tubes. Now let us discuss about companion cells. The function of sieve tubes are controlled by the nucleus of companion cells. Parenchymatous cells which lie on the side of the sieve tubes are known as companion cells, which have cytoplasm of companion cells, and sieve tubes are connected by pith fields present in common longitudinal wall. The cells maintain pressure gradient inside the sieve tubes. Sieve tubes and companion cells develop from the same mother cells, so they are called as sister cells or ontogenetically related cells. Then let us discuss about phloem parenchyma. The elongated cells then have dense cytoplasm and nucleus and thin cell wall made up of cellulose. Then monocot phloem parenchyma. In monocots, the phloem parenchyma is absent. Storage. Food storage like starch and fats, resins, mucilage, latex are stored in phloem parenchyma. Then phloem fibers, which are also known as blast fibers. They are made up of sclerenchymatous cells absent in primary phloem. Phloem fibers are certain plants are used commercially, like jute, then flax and hemp. Now let us discuss about components of phloem in gymnosperms. Instead of sieve tubes, here sieve cells are present, and cluster of sieve cells are known as sieve areas, where we do not have here companion cells. Instead of companion cells, we have albuminous cells, which are linearly attached to the sieve area, the phloem tube. Instead of they provide they act as companion cells and provide the direction to the sieve cells. Now let us discuss about tissue system. There are three types of tissue: epidermal, ground and vascular. First, epidermal tissue system. They are living cells which are parenchymatous in nature, compactly arranged without intercellular spaces. They have a small amount of cytoplasm and large vacuole. In aerial parts, to prevent water loss, they are surrounded by waxy layer known as cuticle, which is made up of waxy substance known as cutin. Generally, epidermis is single layer, but in some plants, to prevent water loss, it is multi-layered. Example, Nerium ficus in leaves, whereas in orchid roots, example is Vanda. Then epidermis of aerial parts usually have pore-like structures called as stomata, where roots do not have stomata. Then stomata have structures, guard cells, subsidiary cells and stomatal aperture. Stomata, let us discuss stomata in dicots. The guard cells are kidney or bean shaped. They are specialized epidermal cells, and guard cells are green in color due to presence of chloroplast. The inner walls are thick and elastic. The outer walls are thin and elastic. These helps in opening and closing of stomata. The subsidiary cells help ground cells to open and close the stomata. Here is the structure of stomata in dicots. Then stomata in monocots. The guard cells are dumbbell bell shaped, specialized epidermal cells with chloroplasts. The outer walls are thin and middle walls are thick, but both are elastic. The guard cells and subsidiary cells are helpful in opening and closing of the stomata. Here is the structure of monocot stomata. Let us discuss about epidermal appendages. There are two types of epidermal appendages known as trichomes and root hair. Trichomes. They are often multicellular, which are cuticinized. They may be branched or unbranched, which may be soft or stiff. It reduces H2O loss, that is water loss, and trichomes may be secretory. Root hair. They are unicellular, cutin layer is absent, and they are unbranched. They may be they are soft, and water and mineral absorption is done by the root hair. Now let us discuss about vascular system. Types of vascular bundles: radial, conjoint and concentric. First is radial. Xylem and phloem occur in the form of separate vascular bundles. They are arranged on different radii, alternating with each other. They are seen in both monocot and dicot roots. Here you can see this is xylem, this is phloem. They are on different radii and arranged alternatively, so it is known as radial vascular bundle. Then conjoint. Here xylem and phloem together constitute vascular bundle, then it is known as conjoint vascular bundle. There are two types, collateral and bicollateral. Here we can see collateral. In collateral, both xylem and phloem are together, and They have only one patch of xylem and phloem. First, in collateral we have two types. First is open type, that means it has cambium. Which is found in dicot root. Whereas the second type is closed type, which is found in monocot stem, which does not have cambium. Then bicollateral, here we can see two patches of phloem and one patch of xylem and two cambia. It is seen in families like Cucurbitaceae, Apollinaceae, Solanaceae. Conjoined collateral closed type of leaves are found in both monocot as well as dicot leaves. Now let us discuss about concentric vascular bundles. On one type of vascular bundles, tissues form core and other surround cells. For example, here two types are there: hydrocentric and leptocentric. Here in hydrocentric, the xylem is surrounded by phloem, and in leptocentric, the phloem is surrounded by xylem, which is amphicribal and it is apivasal. It is seen in ferns. It is seen in yucca. Now let us discuss about monocot root anatomy. Epidermis. The outermost layer bears large number of unicellular root hairs. Cuticle and stomata are absent in this. Then cortex. It is hypodermis is absent. The cortex is made up of parenchymatous cells. Cells are loosely arranged. Endodermis is very distinct, made up of barrel-shaped cells having Casparian strips. Then pericycle. It is also very distinct, and the cells of pericycle give rise to lateral roots only as cambium is absent in monocots. Then vascular bundles. Vascular bundles are greater than 6, so called as polyarch condition. These xylem are in endarched condition. Then conjunctive tissue. They are between the patches of xylem and phloem. The parenchymatous cells are present, which comprise the conjunctive tissue. Pith. Pith is a large and well developed in roots of monocotyledonous plant. Here we can see the structure of monocotyledonous root. Dicot root anatomy. Same as monocot root, but the vascular bundles are about 2 to 6, that is diarch to hexarch condition. The pith is reduced or absent due to excessive formation of metaxylem. Here is the structure of dicot root anatomy. Comparison of dicot and monocot root anatomy. Characters. First is pericycle, then dicot root, monocot. Give rise to secondary root, vascular cambium and quark cambium. Here monocot gives rise to lateral roots only. Then vascular bundles give rise, here dicot give rise to diarch to hexarch 2 to 6. Here monocot polyarch more than 6. Then cambium develops at the time of secondary growth. Here monocot cambium is absent, no secondary growth is seen. Pith. In dicot root, the pith is small and inconspicuous. In monocot root, the pith is large and well developed. Now let us know about dicot stem anatomy. Layers of dicot stem. Epidermis is outermost layer, which is cuticalized, which may form trichomes and few stomata. Young stems may bear stomata. And cortex is several layers, and cells are arranged in multiple layers between the epidermis and pericycle constitute the cortex. Cortex has sublayers. Hypodermis, first is it has 3 to 5 layers of collenchymatous cells in dicot stem. The intercellular spaces are absent, thick at corners due to deposition of cellulose, hemicellulose and pectin. The collenchymatous cells in hypodermis provide mechanical strength to the young stems. Below hypodermis and endodermis, the general cortex is present, which consists of parenchymatous cells. Cells are thin-walled and round in shape. They have intercellular spaces between them. Then endodermis, the innermost layer of cortex, whose cells store starch grains. So it is also known as starch sheet. Then pericycle. It is present on the inner side of endodermis and above the phloem. In between the endodermis and in the phloem, the pericycle is present in the form of patches. These patches of semi-lunar or half moon shape and they comprise of sclerenchymatous cells. Then vascular bundles. Vascular bundles, here endarched xylem is found, and endarched xylem with conjoint and open condition. Then Ring-like arrangement is seen in vascular bundles of dicot stems. Pith or medulla. The central position of the stem is occupied by a large number of parenchyma cells, which comprises the pith. These cells are round in shape and have large intercellular spaces. Then medullary rays or pith rays. The cells of pith present in between the vascular bundles constitute the pith rays, or also called as medullary rays. These pith rays helps in radial conduction of food. Here is the structure of dicot stem. Monocot stem anatomy. Here you can see the structure of monocot stem. Then epidermis is the outermost layer covered by cuticle, may have stomata. The hypodermis is made up of sclerenchyma. Then undifferentiated ground tissue is found, and it is made up of parenchymatous cells. They have actostele and scattered vascular bundles. Endarched xylem and have water containing cavities which are schizolyogenic in origin. Then let us move on to the differences between dicot stem and monocot stem. Ground tissue differentiated into cortex and pit, not differentiated. Then hypodermis, cholenchymatous, sclerenchymatous. Endodermis is single layered, here it is absent. Then in pericycle it may be homogeneous or heterogeneous, here it is absent. In pith it may be at center made up of parenchyma, or here it may be it is absent. Then medullary rays found in between the vascular bundles, here absent. Then stele, it has U stele which is conjoined, collateral and open type. Here it has actostele which is conjoined, collateral and closed type. Now let us discuss about leaf. Leaf has three layers: epidermis, ground tissue or mesophyll cells, then vascular system. First is dorsiventral leaves. They can be distinguished at two sides. The upper is ventral or adaxial surface, and lower is dorsal or basial surface. Mostly seen in dicotyledonous plant leaves. The upper surface has more number of mesophyll cells due to presence of chloroplasts, and the lower surface has low number of mesophyll cells or chloroplasts. Isobilateral leaves, it has equal distribution of mesophyll cells on the both sides, and mostly seen as monocotyledonous plant leaves. Now let us discuss about dicot leaves or dorsiventral leaves. It has epidermis, mesophills and vascular system. Epidermis is made up of parenchyma, the upper is covered by the cuticle, and it may possess stomata in low number. Here we can see transverse section of a dicot leaf. If stomata is on both sides, it is called as amphistomatic, but if it is only on one side, then it is known as hypostomatic. The vascular bundle in leaf are conjoined, collateral and closed. Here mesophyll of these dicot leaves are divided into two types. First is palisade mesophyll, that means they are tightly packed at the upper surface, and we have spongy mesophyll at the lower surface which are loosely packed. Let us discuss about monocot leaves or isobilateral leaves. Here number of stomata are same on the both sides. It is isostomatic, that means both stomata are same on the both sides. Then mesophyll is not differentiated into palisade mesophyll and spongy mesophyll, and it contains bulliform cells on the leaves which are colorless, empty and large on the adaxial epidermis, which help in leaf rolling. It is seen in Poa and Agropygon. Now let us discuss about the characters between dicot and monocot leaves. Then type of leaf, here dorsiventral, isobilateral. Stomata, more in lower epidermis, and here isostomatic, equal stomata. Then mesophyll, the differentiated into palisade mesophyll, spongy mesophyll, and here no differentiation is seen. Then vascular bundles differ in size, and reticulate venation is seen due to reticulate venation, and here it is similar in size due to parallel venation. Then bulliform cells absent, here present particularly in grasses. Now let us discuss about the secondary growth. Growth of roots and stem in girth with help of lateral meristem is known as secondary growth. The most of the dicotyledonous plants exhibit secondary growth. Secondary growth is absent in monocots due to lack of vascular cambium. Gymnosperms, roots and stems show secondary growth. Secondary growth in dicot stem in stellar region. Here medullary rays are made up of parenchyma, that is they are permanent tissue. These permanent tissue undergoes dedifferentiation division to form interfascicular cambium. Joining of interfascicular cambium and intrafascicular cambium form the ring known as vascular cambium ring. Here dedifferentiation. It is an important biological phenomena whereby cells lose its special form or function and regain the capability of division. Then function of vascular cambium ring. It forms cells both towards the inner and outer side, and the cells cut off by the cambial ring towards the pit mature into the secondary xylem, and cell cut off towards the periphery mature into the secondary phloem. And cambium is generally more active on the inner side than the outer side. The amount of secondary xylem produced is more than the secondary phloem. The vascular cambium ring expands towards the periphery. Now let us discuss fate of primary tissue. It is of three types. Primary phloem get crushed, then secondary phloem get crushed, then primary xylem crushed the pit. Due to these events, secondary xylem or wood is increased and primary and secondary phloem get crushed. Crushing of phloem is known as obliteration. Term redifferentiation. It is a phenomenon of formation of secondary permanent tissue with the help of secondary meristem. Now secondary growth is of two types, meristem medullary rays, then wood. Wood is again divided into two types, seasonal and functional. In seasonal we have two woods, or early wood or late wood, and in functional we have hardwood or softwood. First let us discuss about medullary rays. At some places cambium forms a narrow band of parenchyma which passes through the secondary xylem and the secondary phloem in radial direction. These narrow bands are conditioners from secondary xylem to the secondary phloem and are called as secondary medullary rays. Primary and secondary medullary rays conduct food, water and minerals from the center to the periphery of the organs. The secondary medullary rays are also called as vascular rays. Now let us discuss about wood. With wood is first in wood let us discuss about season. According to season there are two types, early wood and late wood. Early wood is also known as spring wood, then late wood is also autumn wood. Activity of cambial ring is variable according to the climate and internal environment of the plant. Characters between spring wood and autumn wood. Season, spring wood, spring season, autumn season, and here amount more less. Then xylem elements wider and longer, narrow and short. Fibers less more. Color light dark. Density is less and here more. Two kinds of wood, that is spring wood and autumn wood, that appear as alternate concentric rings of the light and dark color constitute an annual ring. Hence, one annual ring includes one circle of spring wood and one circle of autumn wood. It is called as annual ring because it represents 1 year of the type span. It is also called growth ring because as the growth keep on taking place, the annual rings keep on increasing in number. The annual ring seen in a cut stem and given an estimate of the age of the tree. The number of annual rings in the oldest part of the tree corresponds to its age. The age of tree can be known by counting the number of these rings in the tiers of leaf. This study is known as dendrochronology. Then function. Here in function we have two types of wood, hardwood and sapwood. Hardwood is also known as duramen, which is dark in color, and non-functional, which is flexible and which is resistant to pests and insects. Then sapwood is also known as alburnum. Then it is light in color and functional. Differences between sapwood and hardwood. Region, periphery, here core. Then color is light and dark. Here water conduction present, here absent. Then deposition absent, and here deposition of gums, resin, tannins, essential oil, etc. are there. Then density is less, here more. Durability is less, here more. That is, they are resistant to pesticides, parasites and insects, etc. Then hardwood have structure called as stylocytes, which are helpful in blocking of xylem vessels and also called as tracheal plugs. Here, this is known as stylocyte cell, which is periphery uh, to the xylem vessel, which forms a balloon-like structure, which blocks the way of this xylem vessel. With increase in age, the hardwood increases and sapwood remains constant. The secondary growth in the dicot stem in extrastellar region, formation of cork cambium. The continuous increase in drift causes rupture of outermost layer, that is epidermis, and outer cortical cells. They need to be replaced to provide protection to the stem. The new protective cell layers are provided by lateral meristem, that is cork cambium. It is also called phellogen, and this meristematic tissue develops usually in the cortex region. Due to its origin outside the stalk, outside the stele, the cork cambium is also called as extrastellar cambium. Phellogen is a couple of layers thick. These are thin walled and helpful and nearly rectangular in shape. The cork cambium has secondary origin. Function of cork cambium. The towards outside, cork cambium forms a corky layer and forms secondary cortex towards inside, which is outer to inner. Here we have three types of cells which are found in cork cambium. Corky layer, which is also known as phellum, which has thick covering of waxy suberin. Then cork cambium, which is also known as phellogen, which has thin walled and rectangular cells. Then secondary cortex is found, which is also known as phellodrum, which has thin cells which are parenchymatous. Bark. All tissues outside the vascular cambium consist bark. Bark equal to secondary phloem plus peridum. Peridum means we know that cork layer, cork cambium and secondary cortex come under peridum. Bark is of two types: early in season, early or soft bark, then end of season, late or hard bark. Then bark is of two types again: ring bark and scaly bark. Ring bark, where cork is formed completely, example betula, and scaly bark, here we can see the cork is formed in a patches, example guava. Now let us discuss about lenticels. The cork cambium produces loosely arranged thin walled parenchymatous cells with numerous intercellular spaces instead of thick walled suberized cork cells. These thin walled cells, which are now produced instead of thick walled cork cells, are complementary cells, and it creates pressure on the epidermis, which soon results in rupture of epidermis and forms opening called lenticels, and these are lens shaped. Lenticels are highly helpful in exchange of gases and transpiration. They occur in most of the woody trees and permit exchange of gases. Lenticels may be present on stem, root or fruit, but they do not occur in leaves. Let us discuss secondary growth in dicot root. Here we have two types: stellar region, vascular cambium, whereas extrastellar region, cork cambium. First, let us discuss about formation of vascular cambium, that is stellar region. The vascular cambium is absent in beginning, develops later at the time of secondary growth, and hence the cambium is completely secondary in origin. Cells of conjunctive tissue below phloem form first cambial strip, whereas cells of pericycle above xylem form second cambial strips. Then first and second cambial strips join together to form vascular cambium. Functions of vascular cambium. This wavy ring is present below the phloem and above the xylem. Cells cut off towards inner side mature into secondary xylem, where cells cut off towards outer side mature into secondary phloem. This wavy ring slowly becomes circular. Now let us know about the formation of cork cambium, that is extrastellar region. Cork cambium is also a meristematic tissue which arises area results of division of the cell of the pericycle. It is secondary in origin. Cork cambium gives rise to peridum, that is protective cell layer to replace the ruptured epidermis and other cortical cells. The activity of cork cambium is in dicot root is similar to the found in the dicot stem, and it produces cork cells on the outer side and secondary cortex on the inner side. Annual rings formation is completely absent in the root.